So good morning, good afternoon, good evening, and welcome to the MedVR Talk Series. We have about 50 educational recordings of talks by leading medical XR researchers and thought leaders working across the fields of medical domains on our website. My name is Stephen Patterson. I'm the Executive Director of MedVR, a nonprofit educational organization in Boston, Massachusetts. We have two goals. One is to connect with the global community of medical XR researchers and innovators to educate and promote research and development in medical XR through talks by and about the journey of the medical XR innovator. Our community includes about 7,500 interdisciplinary people worldwide. And the second goal is to create a network in the Boston medical cluster of medical schools, hospitals, universities, design and engineering departments, medical device companies, professional designers and developers, and investors to accelerate R&D in medical XR. Today, our speakers and moderator are notable in the decades of research into spatial cognition, or otherwise how the brain interprets 3D dimensional space. This is extremely interesting. It's not a large field, however, very important to medical XR in terms of both diagnostics and understanding how the three-dimensional cognition works. But before I introduce our moderator, I would like to talk about upcoming talks just for a moment, so that you might, if you're interested, sign up for them. So I'll put up from our website the upcoming talks. Later on this month, we have medical extended reality for interprocedural electrophysiology and how in an interventionary lab, XR can be used to navigate and improve outcomes. We have a very, very interesting talk on the 3rd of April by the Center of Innovation in Extended Reality, also known as MIXR, which is a joint collaboration between the National Science Foundation, industry, the University of Maryland, the University of Illinois, and the University of Michigan. Very much looking forward to that talk. They're doing all foundational research, the things that need to be solved for the industry to grow. Things like markerless registration, and I'm again looking forward to that. And then on April 18th, we have XR and surgical training, a scientific evaluation of global health applications. Professor Dr. Jag Dandra is a fascinating person who has been working with VR for many years to educate the larger audience of medical professionals throughout the world. And I think it's important as we grow this this field to include the world in it. So I hope I'll put a link in the chat when after I'm finished here, so that you can get to that page and learn more about these talks. One special housekeeping note: you'll note at the bottom of your screen that there is a chat, and the chat is there for to chat with other people in this webinar. The Q&A chat, it should not be confused with that chat. The Q&A chat is where you want to put your questions that occur to you as you listen to the talk. Now I'd like to introduce our moderator, Madeleine Keehner. She's a principal scientist and chief consultant at Brighter Research. Dr. Keehner is a research consultant with expertise in cognitive psychology, learning science, and assessment science. She has a Ph.D. in experimental cognitive psychology from the University of Bristol and has held postdoctoral positions at UCSF and UCSB and faculty positions at Curtin University and the University of Dundee, where she studied spatial cognition and mental spatial reasoning in real-world domains such as surgery, medicine, dentistry, medical education. In her previous role as Director of Cognitive and Learning Sciences at the Educational Testing Service, she has more than 40 peer-reviewed publications and conference presentation spanning topics such as spatial skills in laparoscopic surgery, interactive 3D models for medical education, and digital-based educational assessment. And with that, I'll turn things over to you, Dr. Keehner, and look forward to this talk.
Thank you. Thanks very much. It's really a genuine pleasure for me to be able to introduce and moderate this talk today. I first met Thomas when we briefly overlapped, actually, as postdocs in the same lab at UCSB. We were both in the spatial cognition lab run by Professor Mary Hegarty there. And I remember quite vividly attending his first talk in that department and being so impressed by the way that he brought together brilliant research design, great experimental design from the kind of cognitive sciences with models from neuroscience and the technical capabilities of VR to be able to really tease apart those non-visible cognitive processes that are involved in spatial cognition. And this is, I think, what Steve was referring to when he talked about the value that those things can bring to this field. I think that there's a huge amount of power in his approach. Since that time, since we first met at UCSB those years ago, he has been has had a faculty position at the University of Edinburgh in Scotland at the Centre for Cognitive and Neural Systems. And he is now Professor for Aging, Cognition and Technology at the German Centre for Neurodegenerative Diseases at the University of Magdeburg in Germany. And he's also co-founder of a VR therapies startup, Neo Mento, which has really provided him with a lot of experience on how to bring VR into the clinic. So having that scientific background, I think, and then bringing it to a startup is a really interesting and important journey for this field. Throughout those years, he has organized symposia internationally in the USA, the UK, Portugal, the Netherlands, and Germany. In Germany, he is also chair of the biannual interdisciplinary symposium on spatial cognition in aging and neurodegeneration, ISCAN, which is very strongly related to his work. So please join me now in welcoming Thomas Wolbers to give us this exciting talk on his work. Oh, Thomas, you're muted.
Unmute myself. Yeah, right. Excellent. So thank you very much, Madeleine, for this very kind introduction. I also vividly remember the first talk I gave at UC Santa Barbara. Yeah, so I'm going to talk about our topic that we've been working on for quite some time. And in order to do so, let me share my screen. All right, I hope you can all see this title slide. Yeah, so the topic of today's presentation is, can we measure spatial navigation with extended reality? And could that be something like a unique window into early stages of dementia? So before getting into the topic, just some background. So the center I work at, in German, DZNE, which is a long German name, but as Madeleine said, it's the German Center for Neurodegenerative Diseases. And it's a bit unique in the sense that the center is spread all over Germany. So basically 10 sites that cover different areas of research into neurodegeneration. And I'm actually based here in Magdeburg, which is like halfway between Berlin and Göttingen. So this is what I'm going to talk about. I'm first going to try to make some arguments why it's important to think about and to establish new tools for assessing dementia. And I'm going to have take a strong focus on Alzheimer's dementia. And in doing so, I will try to convince you that spatial navigation could be a very promising route, a very promising tool, a very promising approach in this endeavor to help us develop more sensitive assessment tools. I'm then going to show you some experimental work, both our work, but also work from other groups that have really tried to characterize in detail how early stages of Alzheimer's dementia affect spatial cognition. And I'm going to take a focus on what's termed here path integration. I'll explain later on what that means. And then in the end, I'm going to conclude with a couple of slides on, okay, if we want to now take this research, which at this stage is mainly experimental, into the clinic, what are kind of the challenges that we might be facing, what are also the opportunities that could arise and that await us. And then I'm going to conclude with a little bit of summary and outlook. Right. So let's get started. Just a brief background on Alzheimer's disease. So you might know that Alzheimer's disease is basically a neurodegenerative disorder, and it's characterized by two main neuropathological processes. One is the deposition of so-called beta amyloid plaques and the buildup of neurofibrillary tangles by means of the hyperphosphorylated tau protein. And this then leads to all kinds of things like, as you see here, and overall atrophy. We see deficient synapses and others. And when we look at the behavioral level or the cognitive level, there are a range of cognitive but also behavioral symptoms that can be observed in people with especially more advanced stages of Alzheimer's disease. Obviously, paramount are memory deficits, and in particular long-term memory deficits. But you also see things like visual spatial difficulties, like perceptual difficulties, deficits with planning, reasoning, so-called executive functions. And on the behavioral level, you can often observe things like apathy, signs of aggressive behavior, social withdrawal, and others. So it's quite a mixture of phenomena and of deficits, of changes that can be observed in especially the more advanced stages of Alzheimer's disease. And coming to the topic of the talk, one very common symptom, behavioral symptom, that's also observed in Alzheimer's patients is what has been termed wandering behavior. Yeah, so wandering behavior basically means that when you have a patient who leaves their home independently, so by themselves, then quickly they can become lost. Yeah? So they can become lost, confused. Even after just walking around for maybe a couple of turns, they might get lost. They might have trouble finding their way back to where they came from. And this can actually even have fatal consequences. Yeah? So there are statistics showing that, especially in harsh weather, so when say in winter when it's cold, that if these patients are not found within the first 12 hours, there is actually quite a high likelihood that people might die. So it's actually quite a dangerous phenomenon, a dangerous symptom of Alzheimer's disease. And as I said, it's relatively common. Some statistics say it's the second most common behavior of dementia patients. Right, so far so good. So you might have heard that in recent years, and especially last years, there has been some interesting and relevant progress in developing new therapies for dementia. So last year, the FDA has approved a new drug called lecanemab, which is kind of an antibody therapy that kind of clears or removes amyloid from the brain. And this will now also probably be approved in other countries as well. And one key factor is that in order for this and other therapies that I'm sure we're going to see in the coming years, for them to be maximally effective, it's important that those therapies can start as early as possible. Because if you have already sustained a lot of neurodegeneration, yeah, it will be too late, and you cannot reverse this. You cannot kind of revitalize neurons, for example, that might have died. So the key thing is that we need to identify those patients as early as possible. Yeah? You could now say, okay, well, why don't we know that we see this buildup of amyloid pathology, we see this buildup of tau pathology, why don't we just try to measure it somehow? And the most established methods to do this are either PET, so which is positron emission tomography, or lumbar puncture. You basically extract a cerebrospinal fluid. This works very well, and you see already in early stages of the disease that we show here on the x-axis that there's quite a buildup of this pathology. Yeah. The problem is, though, that those measures are A, highly invasive, and B, they're very, very expensive. So they're unlikely to become part of standard clinical routines, or some part of, say, some annual checkup. Other measures, however, that you can do, such as, for example, looking at structural changes in an MRI scan shown here in this purple curve, or even worse, cognition, shown in this green curve, they show very little changes in those early stages, and only at later stage, here termed MCI, which stands for mild cognitive impairment, you start, for example, for cognition to see also changes there. So those measures that are currently used are not as sensitive as those direct results of pathology, and that obviously makes it tricky to identify, to find those patients in these very early and even in those pre-symptomatic stages. So how can we do better? How can we improve on this situation? And one topic or one function that has been proposed for a number of years now is actually spatial navigation. Yeah? So this is a review paper from 2018, where the authors basically describe the current state of knowledge on, okay, what are the spatial navigational deficits that have been found in early Alzheimer's disease, and do they have any kind of diagnostic or prognostic value? The problem is at this stage that we don't have any standardized clinical assessment tool for navigation. Yeah, we have very nicely validated and psychometrically validated tools for measuring, say, verbal memory, perception, executive function, but for navigation, such tools currently do not exist. Okay. So I'm going to dive one more slide. We're going to talk about the biology of Alzheimer's because it's very relevant to the future of, to the rest of the talk. And this is going to show you why navigation could be particularly sensitive in these early stages. So this is a staging, a figure here that shows kind of the build-up of this tau pathology, so this neurofibrillary tangles across the disease. And those have been classified into different stages. Yeah? And typically, the data suggests that you see a buildup first in the brainstem, and then already at the first cortical site that's being affected is a region that's called entorhinal cortex. Yeah? So it's one of the very, very early regions that is affected by tau pathology. So the idea came up, well, maybe if we can get some kind of functional readout, if we can somehow have an indicator of potential deficits or potential changes in entorhinal cortex, maybe this could help us to see these very early stages, to detect those very early stages of Alzheimer's pathology. And this is now finally where navigation comes in, because, as you might know, in 2014, the Nobel Prize in Physiology/Medicine was awarded to these three scientists, the Mosers, Edward and May-Britt Moser from Norway, and John O'Keefe from UCL in London. And basically what they had done, they had discovered in rodents two cell types. One cell that's called place cells in the hippocampus, and the second one is termed grid cells, and they were found in entorhinal cortex. And what these cells primarily do is that they function as like a GPS system. So basically they code for the animal's position in, for example, a room, in some recording chamber, as the animal walks around. So different cells firing at different locations, and you can precisely reconstruct the position of the animal by recording from a collection of cells. Later on, those same cell types were also described in humans. Yeah. So, and this now brings us to this realm of, okay, we have potentially with these grid cells, we have potentially a function, we have a GPS signal that could be affected in those very early stages of Alzheimer's disease. So we set out in 2018, actually published this study, and to first look at, okay, do we actually see any evidence for changes in grid cell computations as people get older? Yeah. So this was a former PhD student in my lab who now works at Boston University, Matthias Stange, and he actually ran a study in which he put subjects, younger and older adults, in an MRI scanner. But while they were looking at a screen, you can see here, and they were doing kind of a desktop-based navigation task. So you see a first-person view of this environment here. So somebody would be navigating through this virtual environment. This is just 2D on the screen. And then we were looking at, okay, we're measuring signals here bilaterally in entorhinal cortex. So we're checking, okay, what's going on? What do these grid cell signals look like as subjects are doing this task? And what we observed is, that we could replicate findings from previous studies, and that in healthy younger adults, we see a reliable grid cell signal. But this signal was almost absent in most of the older adults, not in all of them, but in many older adults, this signal was absent or was kind of or very noisy. So there was definitely a compromised grid cell signal in those older adults. So far, so good. However, can we also, as I said, can we also have something like a behavioral readout? Can we somehow have an experimental paradigm that tells us how good or bad the functioning or the state of this grid cell system might be? And this is now where I come to this topic of path integration. So path integration is a very simple navigation behavior. And what it means is basically, how good are you at monitoring or measuring self-motion cues in order to update your position in an environment. So this is a very typical way of measuring path integration that has been conducted in many studies, already starting in the 1980s. So what you would do is you would have a participant, like a subject in a study, and you would guide them, for example, along an outward path, like, for example, here, your first kind of translation, you have a turnaround, make a second translation. And then at the end of the second translation, they have to indicate where they think the starting location is. Yeah? And basically, they have to do this via beeline assessment. Yeah? So either they have to walk back to start or maybe point to start. There are different ways of measuring that. Yeah? And usually, when you have a very, say, austere environment, or some studies have also been conducted with people being blindfolded. People make errors. People are not perfect in this, so they might indicate a location that is somehow away from the true starting location, so you can measure their path integration error. This function, this very basic navigation computation of monitoring self-motion cues to keep track of where you are, is supposed to critically rely on this grid cell circuit. There's a lot of animal data suggesting this. And indeed, what we did back then in this study, that we then said, okay, look, we have these older adults here, and we also see there's some variants, some are better, or show a stronger signal than others. And we actually found that the strength of this grid cell signal, as shown here on the y-axis, was related to their performance in the path integration assessment. So meaning that those older adults who were particularly poor at doing such a path integration task also had almost absent grid cell-like signal, so showed a very compromised grid cell signal. So that's already reassuring, because it tells us that maybe path integration really could be a sensitive readout of these entorhinal computations. So next step, if that's correct, then maybe we can use path integration as kind of a sensitive readout of emerging Alzheimer pathology. Yeah? So maybe people who start to develop these amyloid plaques and this tau pathology, those neurofibrillary tangles, maybe they already show deficits and more severe deficits in path integration. So is that correct? And the question is, okay, what patients do you look at? Yeah, again, we're going back here to this figure of progression of Alzheimer's pathology. And an interesting stage is this stage here, which is termed as MCI, so mild cognitive impairment. So this is supposed to be a prodromal stage of AD, meaning that many people who are classified as having MCI go on to develop full-blown Alzheimer's disease. And what you find in these people, and this is the definition, is basically that they already show abnormal cognitive decline. For example, in a memory test, when you run some standard memory assessment, so they perform much poorer than age-matched controls. But it's not as severe in terms of impacting on daily life, so they can still live independent lives. But they clearly have an increased risk of going on this trajectory to then ultimately develop Alzheimer's disease. So this is a study from a group at Cambridge University in the UK, and they then said, okay, let's take a group of MCI patients and let's see how they perform in an immersive path integration task. So you see the setup here, subject will be wearing a head-mounted display, and this is the pathway that they were walking along, so this kind of triangular shaped pathway, and then in the end, the subject was supposed to go back to this starting location. They did this in different environments. We don't have to go into the details here, but I want to highlight two things here. First of all, compared to healthy controls shown here, this path integration error shown on the y-axis was higher, significantly higher in those MCI patients. Even though these are controls that are matched on age, gender, education, and other things, but clearly those patients already showed a higher or error or worse path integration performance. Secondly, they also performed a lumbar puncture on these patients. So they basically extracted cerebrospinal fluid and then looked at, okay, how much amyloid, for example, is there already in the CSF of these patients? And there are established thresholds where you say someone has already developed a significant amount of pathology. So they kind of stratified this patient group into what the term MCI negative, so those below the threshold, and MCI positive, those above the threshold. And indeed, you see quite a striking difference here in that those who had already built up quite a significant amount of pathology also performed much worse on this path integration test compared to those where pathology was still relatively minor. And critically, they then ran an analysis where they tried to distinguish biomarker positive from biomarker negative MCI patients and compared this to standard clinical tests. So to tests that you would be administered to if you were a patient and you would go to see a neurologist or psychiatrist in a clinic. And these are kind of standard commonly used tests for dementia assessment or AD assessment. And it turned out that this kind of classification accuracy or the diagnostic accuracy of this path integration task was significantly higher compared to those currently used tests. So this is very reassuring because it tells us that it seems like a VR-based assessment of path integration could be a more sensitive measure for my cognitive impairment status than currently used cognitive tests. Okay, so far so good. So we've now talked about the MCI stage, and that looks at path integration is more sensitive than common measures. But what about, because I said earlier that we want to try to initiate potential therapies as early as possible. What about even earlier? What about this kind of pre-symptomatic stage where in pathology there is already a buildup, but clearly, for example, cognition, there's nothing where you don't see any deficits whatsoever. So there's a group of people that have been termed who experience what has been termed subjective cognitive decline. And this basically means that these are people who report that they feel their memory is fading, they feel their memory is getting worse and worse, but when you test them on standard cognitive tests, you see no impairment. So they perform within the normal range of age-matched controls. However, there's data showing that you seem to see already a significant buildup of tau pathology, especially in entorhinal cortex. And it also looks that many of these patients actually, again, have an increased risk of going on to develop Alzheimer's disease. So it is believed that this could be the first symptomatic manifestation of Alzheimer's disease, even though you currently cannot verify, you cannot show it with standard cognitive tests. So in my lab, Vlada Segan, who's a postdoc, set out to look at this, and she basically developed a paradigm that's kind of similar to what I'd shown in this previous study. Subjects would be wearing a head-mounted display, and they would be walking along trajectories. In this study, we used curved trajectories for various reasons. And interesting, we tried to also have a self-guided task. Yeah, so we basically had like a virtual object in this case here that would be flying, and your task was to kind of follow this object, and thereby you would be guided along this trajectory. And then when you reach the endpoint, your task was again to indicate the location where you came from on a beeline. Vlada also conducted standard cognitive testing and showed, as expected, that those SCD patients, so those patients with subjective cognitive decline, were on par with matched controls, so did not perform any poorer on standard memory, attention, executive function tests. However, when she looked at path integration error in this task, she already saw that even those STD patients seemed to perform poorer, so basically showed higher path integration error compared to the control group. And secondly, she also obtained blood samples of these patients, or of the entire group, and we analyzed what's called a neurofilament light chain. It's basically a protein that's released into blood, but also into the CSF, when there is neuronal damage. So it could be some kind of marker already of ongoing neuronal damage. And it turned out that there was a relationship that those who had more neurofilament light chain in the blood also showed higher path integration errors. So this is promising, and then it seems to suggest that maybe even in these very early asymptomatic or pre-symptomatic stages, we can already start to get a measure of cognition, in this case of path integration, that again is more sensitive than standard tests. So this is now fundamental research that I've told you about, and but what if you want to take this now to a clinic? Let's say we're not the only ones, and there's other groups working in this field. So let's say that the evidence is more and more converging, that this works well, that you can identify people. But of course, this is at this stage experimental research. So what if we want to bring this to some kind of clinical setting? What if we want to turn this into a clinical assessment? So I want to first talk about the opportunities. And I think, to me, one major opportunity here is that path integration, it's such a simple behavior. As I said, you just walk a very simple trajectory, that it's highly likely that this could be a very good tool for kind of a culture-free assessment. It's not so much, probably not so much biased by cultural experience as other tests are. Secondly, it could provide a very suitable tool for things like cohort studies, also multi-site or multi-center studies. So if you have multiple sites that jointly conduct a study, but in the end, hopefully also for routine clinical assessment. Third, I think it could also be very helpful for translational research across species. So don't forget, you might be aware that, for example, in neurodegenerative disorders, a lot of this preclinical research is done on non-human animals. So often, let's say, rodents. And then to make this translation from the animal model to humans, it's often difficult to say, okay, what is a comparable behavior? How can I use a task that's been established for rodents, for example, and how can I adapt it to humans? And this path integration behavior is behavior that has also been studied extensively in rodents, so it could really help us improve and make this translation from the animal model to the human case. And finally, I think it could also be very interesting and helpful for clinical trials. So basically, when you have a new treatment, and you want to measure how effective is this treatment, it might well be that kind of path integration assessment gives you a more sensitive readout of therapeutic effects compared to currently used measures. So this, I think, could be another important benefit. However, in doing so, when you think about bringing this to the clinic, there are also a number of challenges that we'll probably have to overcome, and I'm just listing a couple of them here. So first question is, okay, many of these subjects will probably have zero experience with extended reality. So we're talking people 60, 70 plus. So most of the subjects that we tested, for example, in our studies, have never had any experience with VR before. And we needed to familiarize them first with the technology and how it feels like to be immersed in VR, how you interact with the virtual environment. So if you think of, say, a high-paced clinical diagnostic situation where time, for example, is at a premium, and maybe there's not that much time to familiarize someone, could this very unfamiliar testing situation maybe evoke anxiety and thereby impair performance in some people? So could it maybe confound your readout? Secondly, in most experimental studies that have been conducted, I would say they're relatively lengthy, in the sense that people do many, many trials to get a precise estimate, a precise measure of people's path integration abilities. But in the clinical setting, obviously, this is often difficult. You want to have a diagnostic assessment in relatively short amount of time, while still acquiring enough data. So how can you find this compromise? Third, I've mentioned that many or most cognitive tests that are in use in clinical settings also have been validated extensively and there have been big studies to establish, for example, age norms. So how would a typical 65-year-old person perform on this test? How would a typical 70-year-old person perform on this test? So you need some kind of comparison values. And this is, in a sense, easy because those tests, for many of those tests are things like paper pencil tests, and they haven't changed in the last 30, 40 years. It's the same way of administering this test. However, we're talking about XR, we're talking about a development where the hardware is also constantly changing. So the headsets that we are using today are already very different from what we've been using for just even 5 years ago. So how can we establish age norms that are consistent and that are valid, even though the technology and the hardware might change? And then finally, a critical factor is also that you may be aware, maybe from personal experience, that navigation abilities is a function where typically we see a lot of inter-individual variability. So there are some people who are extremely good at navigating, who are really expert navigators, and whereas there are others for whom this is much more challenging. So how can we account for those baseline differences? And what approaches could we take to make sure that we detect people very early on if they start to develop a disease, in this case, Alzheimer's? Right, and this brings me to the very last slide of this presentation. So I hope to convince you that path integration is a very simple navigational behavior, much simpler than many other functions that have been tested, but it's strongly dependent on grid cell computations in entorhinal cortex. And there's converging evidence, both across labs, but also across species, that path integration is compromised in early stages of Alzheimer's disease. And by species, I'm mainly referring here to rodents, because there are rodent models of Alzheimer's disease. And also in these models, you find that grid cells are compromised early on in the disease, and secondly, that this seems to have an impact on path integration behavior. So it's very nice that we see this convergence across species and also labs. Third, there are studies that I haven't mentioned that have shown that people in even younger adults, so people say below 50, already can show path integration deficits if they carry some kind of genetic mutations that increase the risk for developing Alzheimer's disease later on in life. You probably know that there are certain genetic risk factors for Alzheimer's disease, and it turns out and in those patients or those people, you can already identify path integration. It's very slight, very minor, but they seem to be there, path integration deficits. So another argument to support this point that path integration might be a very sensitive tool for AD detection. In bringing this to the clinic, I think ease of use, both for patients and for clinicians, will be essential. In many cases, XR still is a technology, as I said, that people are unfamiliar with and that requires some kind of technical expertise. So I think it's really fundamental that we try to find solutions and setups and testing scenarios here that are very easy to use, both for the patient, but also for clinicians. Likewise, technology, as I said, evolves, and I think we should try everything. We should try everything we can to further simplify VR setups. So in the studies that I've shown you, for example, and these were studies that still involved like an external tracking system that were used to track user movements. And this is just some part of the technology that obviously that can fail, that it can be difficult to troubleshoot. So anything we can do to try to simplify those VR setups, we should consider and we should explore. And finally, I think it will be imperative to also validate these measures in longitudinal studies to assess how predictive it really is. All the data I've shown you today are from studies that are cross-sectional, so you do like a one-off measurement, and you then try to distinguish between patient groups or between patient and controls. But in the end, what you want is you want to also track people over time. You want to check, okay, how predictive is this path integration assessment for a patient or a person's future trajectory? So will those that really perform poorly on these path integration tests actually really go on to then develop full-blown Alzheimer's disease? So validation and longitudinal studies will also be essential here. Yeah, and with that, I've reached the end of this talk. I hope it was clear and understandable, and I'm very happy to take any questions you might have. Thank you.
Thank you so much, Thomas. That was fascinating, and lots of interest and lots of comments in the chat. People saying how fascinating it was. And just a reminder that if you want to pose a question, and I see a couple already, please use the Q&A chat bubbles at the bottom of your screen rather than the chat. So one of the things that I think that occurred to me listening to what you were saying is that this is such a great example of how fundamental science and how findings from basic science can be so valuable and so informative in ways that you only get really by bringing together those decades and decades of research and all those kind of careful findings. I'm thinking about the way that you were able to identify those grid cells in the entorhinal cortex as being that really early indicator that does a better job and a better job than other sort of non-invasive standardized measures, and it can detect it at such an early stage. And for me, that's a great example of how the kind of, you don't know what you don't know, but there's so much foundational research that has been done that gives us insights into what's going on under the hood, that I feel that I want to kind of say to people who are in this industry and who are building these kinds of applications, talk to scientists, find out, because there may be things that have been discovered, that have been learned about in science, sort of published in journals that maybe not that many people read outside of those fields, that are actually potentially really, really valuable and could add that extra dimension of precision and kind of predictability and so on. So that's my kind of that's my soapbox, really, is talk to scientists, get scientists involved. So
yeah, can I just say something to that? No, I absolutely fully agree, but I think it really goes both ways, right? It's like for people in the industry try to talk to scientists, but on the other hand, maybe scientists somehow, and I would include myself here, have a tendency to also stay within their own community, maybe go to scientific conferences and not seek as much contact as we maybe should be seeking to people who work also in maybe non-academic contexts or industrial contexts. So I think we as a scientist are also obliged to kind of try to seek those opportunities. And for me, this is one of them. And that's why I'm always happy to also take part in these things.
Fantastic point. Yes, and actually it's really nice to see some of those conferences now really growing, which bring together the science and the industry, and often science, industry and medicine, which is a really powerful combination. So if I'm thinking about this, using this in real world settings of clinics, for example, if a clinician was to say to you, this is great, I can totally see this makes a lot of sense, it's non-invasive, I can do this, but I can do this without a headset, without VR, without XR, what would you say to that? I mean, does the XR bring something that is particular, unique and necessary to this?
Yeah. Yeah, I mean, it's obviously a challenge to say. Ideally, people want to have maybe even a smartphone app or something you can run on a tablet or laptop to do this much easier. And there are people who are working on this. Yeah. So I would say at this stage, the jury is not out whether or not it's going to work well or not. However, there are some considerations, some things I think might make me a little bit cautious. One is that, as I tried to indicate, path integration is a behavior that, in a sense, is multisensory. Yeah, so it involves not only vision, but it also involves like sensing your vestibular cues, like about acceleration, about turning, stuff like that. It involves like proprioception. So if you just show someone, say, a smartphone animation or a laptop animation of a movement through via some trajectory, you lose these cues. And the question is, would you still be as sensitive as you currently are? Secondly, I don't think we should underestimate the factor of field of view. Because in VR, we can talk about, in XR, we can talk about 100+ degrees of field of view now. If you look on a small laptop, obviously we have a much smaller field of view, and you can even have discrepancies between a simulated field of view and the actual field of view of the user. Third, I would say that you even start to induce a sensory conflict, because you now have a visual information that tells you, oh, you're now moving across this trajectory, but your body tells you, oh, wait, you're stationary, you're not moving at all. So as a patient or as a subject, you would have to overcome this this conflict and basically say, okay, I'm going to ignore my body-based cues and just follow what vision tells me. So this is something that might also require more training, but I remember that there are experimental studies that have even shown that sometimes in those desktop studies, you can even have people who have trouble updating their orientation. So you see a trajectory which would be changing your orientation, but when you ask them to respond, they would respond as if that orientation had not changed. So at this stage, I think I would say I'm cautious, but of course the jury is out whether eventually we might find solutions that work.
Okay, and I think what you've said is chimes with something that Steve said in the introduction, which is that the three-dimensional nature of XR and in this case VR brings something to a person's mental representation that is different, is qualitatively different, and that we operate in a three-dimensional world, in the real world. We evolved for that, and so that difference between what you get with a 2D or static representation versus an immersive 3D representation is really, really different in terms of what your brain is doing with that information. There's been some interest in the chat and also in the Q&A with about other types of data, in particular eye movements, eye tracking data. John Hartman has asked whether you've done any work with eye tracking during in these path integration studies, and some people have talked about haptics, and you just mentioned proprioception, this multimodal, our representation in our brains. We talk about spatial cognition, not strictly visual cognition, because those two things overlap, of course, but spatial is a little more multimodal. So can you talk about that? Are there other forms of data that you would like to introduce? Have you worked with other forms of data, other modalities?
Yeah, absolutely. So we haven't done it yet. So eye tracking for sure is something we definitely want to see, we definitely want to explore, and we already have some projects in the pipeline that would also include eye tracking. Beyond that, I think also gait could be really interesting, because there's a lot of research suggesting that, for example, cognitive deficits can also lead to alterations of gait behavior, in particular in situations where you're cognitively challenged, which you are in this situation. And similarly, head scanning behavior. Yeah? So how people actually move their head as they walk along these trajectories could also be something. And there's some experimental work, I remember, that also showed alteration in head scanning depending on cognitive load, for example. So yeah, these are definitely parameters would be very interesting. And I would say eye tracking also, in the sense, not only looking at, okay, where do people look, what's their scanning behavior, what's their saccadic behavior, but also maybe looking at things like pupil dilation. Yeah, because there are also attempts to show that pupil dilation could be a very sensitive indicator of stress, cognitive load, and maybe these are also parameters that, if you combine them with those behavioral readouts that we currently have, your diagnostic accuracy might even further improve.
Fantastic. Yes, great. Actually, I love the idea about looking at gait, and in fact, that did come up in one of the questions. So there's your answer. The person who asked that, who's anonymous, that yeah, external sensors looking at things like gait and movement, so on. That's.
But I just want to again make this one argument that I made before. So as I've also been also involved in this VR therapy startup, which has kind of a different angle. Again, what we've learned is really that ease of use is fundamental. Yeah. You can have situations in clinical context where people will end up not using the technology because it's just way too cumbersome to set all this stuff up. Yeah. So if you now say, okay, let's do gait, and let's also maybe attach trackers, body trackers to various body parts, it's a slippery slope, and you can get into this terrain where a lot of people then just not use it, because it's just too cumbersome, too difficult, too prone to fail also.
Fantastic. Actually, that's a really important point, that translation from the basic science to the applied world, where people are busy and they have resources and they need information that's not onerous and to get and so on. Okay. There was a question from the audience about whether certain visual conditions, for example, glaucoma or I'm not quite sure how to pronounce this, amblyphobia, I think, or I'm not sure I'm saying that right, whether that would prevent those kinds of conditions might get in the way of being able to use this kind of assessment. Do you know the answer to that?
So I would say it can confound it. Yeah, so there's a little bit of research on age-related visual disorders. So remember, like also macular degeneration is a condition that has been investigated, and it seems that this can affect your behavior. Yeah, and basically because you're not getting all the information that a normal, healthy, or like visually healthy user would get. So you're getting less optic flow, for example. And that could basically lead to further impairments in performance. Yeah. So if you have these special populations, maybe you would need to develop disease-specific normative data to see how such population, or you would need some control for the extent of the damage, because typically, obviously, this varies across patients. But they can use it. Yeah, they can use it. We've actually run a study in collaboration with some ophthalmologists in our lab where we had people with macular degeneration. They could do it. They certainly could do the task, but their performance was clearly worse compared to controls. Yeah.
Okay, great. That's kind of thing is really important to know. So there's an interesting question about the numbers and types of visual cues, and this is something that I was interested in also in asking about. When you're designing a VR environment, when you're trying to get that get the simulated environment such that you will get really clear signals, you better sort of extract the signal from the noise and reduce the measurement error. What are the kinds of things what are the kind of steps you had to go through, and what are the kinds of outcomes of that learning process?
You mean in terms of like designing the environment and designing the task? Or?
Exactly.
Yeah. So first of all, as I showed in this study that we did, currently the types of environments we're using are very, very simple. Basically, they just, for example, consist of like a ground plane that give you optic flow information, nothing else. Because the theory would say path integration is a behavior that is based on self-motion cues. You don't want to have like static objects like a tree or whatever that kind of you can also use to derive your position, but just kind of giving you self-motion cues. However, more recent studies suggest that even in those richer situations, it might work. You could still see these kind of diagnostic benefits of path integration. So we could probably move to also more realistic or more ecologically valid environments and still show these deficits / still have like some diagnostic benefit. What we found is that yes, an experimental study, we have time. And we can spend lots of time training people, making sure they can handle the technology well. But we're now currently doing first steps towards more clinical settings. And this is when we said, okay, we have to we have to involve people who are experts in things like user interface design, user experience design. So we've actually worked with the design studio to completely change and streamline and simplify this whole onboarding process of how someone who's never experienced VR will be able to quickly learn what to do and how to behave in this particular setting. Yeah, and this really for us was quite a learning experience and was very, very helpful. So we've now really simplified the whole way of instructing people. It's not like multimodal instructions that we give to people. It's a lot working with icons and images and stuff like that. And this really has made the whole experience much easier for patients/subjects. We also needed to make sure, because you can think of this like when you measure this behavior, that it's kind of a representation of position that's somewhat noisy. So if you read out position knowledge once, you will get some measure. If you read it out again, you will get a different measure. So this kind of variability across trials that you're getting when you repeatedly test them in the same trajectory also tells you something. It tells you something about how noisy this knowledge is, which can also be very informative. Yeah? So this is also something we've learned along the way, this kind of trying to distinguish between what's called accuracy and precision could also be very helpful and fully characterizing these patients.
Super. And so how many trials do people typically need to do for you to get good data, to get a good signal?
It's a constantly shifting metric, which, as I said, we're trying to reduce it. So at the moment, I think in the latest version we're talking about something like, I want to say 30 something trials. But keep in mind that each of those trials, depending on the length of the trajectory, depending on how you give the response, can easily take like 30 seconds, 40 seconds, depending on how quickly it works. So it adds up. It adds up. So we're currently seeing now how much can you reduce it while still preserving this kind of diagnostic information that we want to get.
I think that's such a nice thing about path integration task. It's so simple and quick to administer. So people have asked about different technologies that we're seeing. Oh, actually, one thing I wanted to say about your previous comment is that learning experience of how to introduce people to the affordances of XR technologies in ways that are effective and relatively quick could be so valuable. Even for example, in medical XR, there's discussions around how do we train people who are going to use this in practice, the actual practitioners too. So I think that getting people up to speed and bringing in some of those principles from learning science, perhaps in terms of presenting that information in ways that people can really get that fluency as quickly and efficiently as possible is a great it could be a really great contribution. Well, outside of this field,
I think it could maybe add one point here, and these are also things we're currently experimenting with in a different context, is to say, okay, what about things like storytelling? What about things like gamification? Yeah, so are there other dimensions that you could use, that you could exploit to really get people in the flow of kind of doing this and quickly understanding and relating to what this? These are things like they're rarely explored, I would say, in fundamental cognitive science or cognitive neuroscience. But I think they can be very powerful. Yeah. And we are starting to work on this. We're not very experienced, but I think it's very interesting. And there are others who have done this successfully already. And I think there's a lot of potential there as well.
A couple of comments in the Q&A about technologies for inducing the experience of motion, for example, multidimensional treadmills, using controllers for apparent motion of self through the environment. Have you had any thoughts on those, or are you aiming to replicate real physical motion, which is actually what people are doing currently?
Right. Yeah, so we have actually also, so first coming to treadmills, your first suggestion idea, we have actually experimented or looked at various treadmills. I guess the challenges that we've been facing so far is that the walking behavior, the way you interact with these treadmills is still a bit artificial. It's still a bit like it doesn't feel like perfectly natural walking. And then if you have people, say in their 70s, 80s, and then they put on a headset so they can't even see their body, a lot of them can feel very insecure, walking on some kind of weird device. And then they might be afraid of falling and stuff like that. So it's questionable, I would say, how precisely, how reliably you can then measure something like pattern recognition behavior if the devices per se is already introducing some additional challenge. In terms of doing it with the controllers, yes, could be possible. We haven't done it, or we did it in one study, it worked, but we haven't explored it extensively. I would say again, it introduces the sensory conflict between what you're seeing and what your body's telling you. So something would have to be overcome. And of course, you might be introducing this additional challenge of motion sickness, right? And we've seen this in our kind of more therapeutic software, where there certainly are users, even if you give them a very slow movement, who can start to feel uneasy. And then we had to do things like teleporting people between locations, which obviously something you could not do here because people need to move. That's kind of part of the task. And so I'm a bit cautious on like using controllers there.
I'm with you completely. I think what we always say in assessment is don't introduce technology for technology's sake. It's only if it's going to make the task better, the measurement better. But if it's led by the technology, that doesn't make sense. You need to lead from the goals and the research and the sort of theoretical grounding, and then decide what is the best fit for that. So I'm completely on board.
Maybe just one point to add to that is obviously, okay, we are, of course, aware that we cannot expect, say, a clinician, a clinical setting, to say, okay, now here you have this kind of, I don't know, 10 by 20 meter superb tracking laboratory where we can do these fancy path integration assessments. So what we're trying to do is actually really kind of scale down those paths. So we're really trying to see how short, how compressed can we make those paths while still showing or still observing those behavioral deficits that we're expecting to see. So that in the end, you could potentially do it in some kind of, say, standard office space, something like that, if it's empty. So that's kind of the approach we're currently taking to make it more feasible in more constrained settings.
Excellent. And that makes a lot of sense. And that's kind of that's one of the processes of doing this translation from science to application, right? Is making it doable. So just we're kind of at time, but let's just have a last sort of input from you on what's next for Neo Mento, what's next for this research, and where you see this going.
Okay, so for this research, as I said, at this stage, we want to validate it in also things like longitudinal studies, which I think is very important. And then we're taking those first steps to make that really ready for things like multi-center studies, so that it could be deployed remotely and people without much training could run it in a very constrained clinical study, clinical trial studies. These are things that I think would be very important, and these are things that we are working on. For Neo Mento, obviously, the focus is a bit different in that it mainly focuses on psychiatric disorders and like more therapeutic approaches. So that's kind of a separate development there. But I think this stuff here on path integration is something we also, as I said, not the only ones working on this. There are other groups as well. And we had a meeting, this ISCAN meeting in December last year, where there was a lot of kind of interest and a lot of activity and a lot of excitement about how can we actually develop this further into making it clinically useful. And I think these are the next crucial steps that we should be taking.
Fantastic. That sounds really exciting. So congratulations on all of this. And we'll pass it back to Steve now.
Yeah, well, thank you very much, Dr. Keehner and Dr. Wolbers. This has been a really wonderful talk, and I think it'll be referenced many times as a recording afterwards. I remember over a year ago talking to Dr. Wolbers about his research in this area and anticipating getting to the point where we could have a talk on it. And the merger of the potential for diagnostics with XR is incredibly interesting. So thank you for making this possible. It's a great talk. And thank you to our audience that came from all over the world. We had people in Asia up late at night across Europe and across North America. And we'll see you next time. Please follow us on whatever social media or register on our website so you can hear about upcoming talks. Thank you.
Thank you very much. I also enjoyed this.
Thank you.